Welcome back to our science podcast. Today, we're diving into a topic that is all around us, yet goes unseen: enzymes. Just imagine, they're the unsung heroes of our biological processes. But what exactly are they?
Absolutely. Enzymes are fascinating. To put it simply, they are biological catalysts made predominantly of proteins. Their main job? To speed up chemical reactions without being consumed in the process. Isn't it incredible how these tiny molecules can have such a massive impact on processes in our bodies and in nature?
For sure. And just to clarify, a catalyst is a substance that increases the rate of a chemical reaction. So enzymes help to make reactions happen faster, a vital function because many biochemical reactions occur too slowly without enzymes. They allow us to live our lives at a much faster pace.
That's right. So, let's dive into some key terminology. The specific molecule that an enzyme acts on is called its substrate. Think of it as the puzzle piece that fits perfectly into the active site of an enzyme. This active site is where the magic happens, as the enzyme interacts with the substrate to form a product. For instance, if the substrate is starch, the enzyme amylase will break it down into maltose.
And speaking of magic, there's something crucial to remember: specificity. Each enzyme is tailored to one specific substrate, much like how a key only fits into its particular lock. This specificity is a key part of how enzymes function in living organisms.
Exactly. And enzymes can also be sensitive to changes in their environment. Two major factors we'll discuss are temperature and pH. Each enzyme has an optimum temperature and pH at which it functions most effectively. For example, most human enzymes have an optimum temperature around 37 degrees Celsius. But did you know that plant enzymes often prefer about 30 degrees Celsius?
Yes, and let's unpack that a bit. When enzymes are exposed to temperatures or pH levels that are outside their optimal range, they can become denatured. This means they lose their unique three-dimensional structure and consequently their ability to function. It's like a perfectly folded origami piece getting crumpled up, no longer able to hold its shape or do its job.
What a great analogy. Speaking of demonstrating these ideas, let's talk about some experiments we can conduct to observe enzymes in action. One classic experiment investigates the effect of temperature on enzyme activity, using catalase found in celery to break down hydrogen peroxide. This is a great visual because we can measure the oxygen released as foam.
That's a fun project. If we were to set this up, we would vary the temperature of the hydrogen peroxide while keeping the pH stable. We'd want to keep careful track of how much foam, representing the oxygen produced, appears at each temperature. It's exciting to see the bubbles grow.
And we can do a similar experiment to view how pH affects enzyme activity too. We can test catalase at different pH levels, say at pH 4, pH 7, and pH 10, which reflects acidic to more basic conditions. The goal is to see how these varying environments impact the enzyme's ability to produce oxygen from hydrogen peroxide. It's wonderful science and allows for real-time data gathering.
Absolutely riveting. And may I say, the visuals from these experiments are worth their weight in gold, particularly when we notice how much more foam is produced at the optimum pH compared to extreme values. It really highlights just how critical pH is to enzyme function. Very exciting. But we should remind our listeners, too, that not all enzymes function in the same optimal conditions.
That's a good point. Different environments can suit different enzymes. For instance, pepsin, an enzyme that breaks down proteins, prefers a highly acidic environment in the stomach with a pH around 2. It's specialized for our digestive processes. This versatility is what makes enzymes key players in countless biological functions.
You know, all of this just showcases how finely tuned biological processes are. Enzymes are a part of the larger picture of metabolism, which refers to all the chemical reactions happening in a living organism. They ensure that reactions happen efficiently and at the right rate. It's almost like having a well-oiled machine running behind the scenes.
Well put. In closing, we must remember that enzymes, while fundamentally microscopic, are essential for life as we know it. They not only accelerate biochemical reactions, but also ensure that these reactions can occur under the right conditions, maintaining our life systems. Understanding enzymes opens a window to understanding life itself.
That's a wonderful thought. So whether it's the extraction of energy from food, breaking down waste, or even the processes that contribute to plant growth, enzymes are indeed at the heart of everything. Thank you for joining us as we explored the hidden world of enzymes today. Keep pondering this wonderful science, and we'll see you next time.